alone, natural killer cell alone, they will all work together, and whoever is successful in killing the bacteria, good for the body. Do you think there is a non-lymphocyte cell that can do the same job, that can kill by the same ADCC? Very good. So there are eosinophils, macrophages, and neutrophils. They all can do ADCC, but the macrophage and neutrophils, they are more importantly, they phagocytose and they kill. We will see now how macrophage is going to do the killing. Yes? Class switching. So the B cell, it recognized this bacteria. The B cell recognized this bacteria. The CD4 cell recognized the same bacteria in the lymph node, okay? But with, of course, with the help of dendritic cell. It was activated, it came into the tissues. The B cell already recognized it because it has got B cell receptors. Now this B cell and CD4, they will directly contact each other. This B cell will say, I have received this bacteria, can I start class switching? It will ask the question from CD4, and CD4 will answer with the help of interferon gamma that yes, you can now start producing IgG instead of IgM. That's why CD4 is called helper. I told you that cytokines will talk to each other with the help of these cytokines, helps in communication of the cells. So CD4 will tell B cell, yes, start producing IgG instead of IgM.
The complement system can do optimization of natural killer cells, or only for natural killer cells, only IgG can do optimization?
No, very good. Actually, opsonization, if you look at, there is a graph that I have not shown you, I think. Normally, complement system can do opsonization alone if on a scale of 1 to 10 we talk. So the opsonization will go up to 5, okay? Or let's say in percentage, it will do 50% opsonization. If antibodies start to do opsonization, that will also do 50%. But if complement and antibody together are in the system, they will result in 100% opsonization. So first thing is that antibodies and complement, they do opsonization collectively in the system. That's more effective, stronger. That's number one. Number two, coming to your question, the complement system helps macrophages and neutrophils more. They don't help too much natural killer cell. That's why it is not, the name of this activation is not antibody and complement mediated cytotoxicity. It is termed as antibody mediated because this is, I just linked it, it's not pure opsonization, it's helping in cytotoxicity. Opsonization is mainly for phagocytosis, for engulfing. Now, with this same point, there is another, this question, is there another non-lymphocyte cell? So yes, the non-lymphocyte cell is macrophage, neutrophil, and eosinophil. But if you look, eosinophil is mainly against parasites, and parasites are large in size. Macrophages and neutrophils cannot engulf the parasites. That's why there is another cell for killing parasites, and that is called as eosinophils. And how do they do it? Exactly like natural killer cell. So now assume this is a parasite, a long parasite, antibodies has bound to it. When eosinophil come, they will recognize through FC receptor and release its perforins and granzymes that will directly damage the parasite. So because it's big, it cannot be opsonized, it cannot be, it cannot be engulfed. That's why these eosinophils have to release perforins and granzymes. So there are two types of strong antibody mediated cell cytotoxicity. One is by a natural killer cell for viral infections or tumors or intracellular bacteria. Other is by eosinophils for parasites. Now a little bit more about CD4 cells. So we have already seen that they are performing all these different helping functions. Now in the bigger picture, so the first part you can see that the antigen presenting cell has presented the antigen. Dendritic cell present the antigen to CD4. CD4 is activated, proliferation take place, and it is differentiated into TH1. Which cytokine is needed for TH1? IL-2 is for proliferation, for multiplication, but I am talking about differentiation. IL-12, just add one to it. Okay, proliferation is number of army is increased, but which type? TH1, TH2, TH17, that depends on special, we call it signature cytokines. So the signature cytokine for TH1 is IL-12. So once IL-12 comes, we have now T helper cells, CD4 cells. Now this CD4 cell can perform lot of functions, lot of helping functions. Number one, macrophages. So here I told you macrophage will engulf the bacteria but will not be able to kill it. Why? So it engulfs the bacteria, phagosome is formed. Inside the macrophage is a lysosome. The phagosome-lysosome fusion, which result in phagolysosome, it will not take place automatically. They will wait. Till when? Till the CD4 cell start producing interferon gamma. When this will communicate, now it will produce interferon gamma. The phagosome lysosome will join. Reactive oxygen species and nitric oxide, hydrogen oxide, they all will be produced. They will kill the bacteria. So that's number one, macrophage activation. Then what else it can do? Complement binding and opsonization. So also the CD4 cells, they help the B cells in class switching, and those antibodies can activate classical pathway. So that's why they can trigger opsonization and phagocytosis. Then if you remember, the first line of innate immune cell was neutrophil. That automatically comes in the first 24 hours, engulf the bacteria and kill. But its killing abilities, like macrophage, can also be enhanced. It can also be increased by interferon gamma. So basically, if you remember, CD4 cell can produce mainly interferon gamma. Interferon gamma will tell macrophage start killing, it will tell B cell start class switching, it will tell CD8 cell killing. So just giving the orders, helping them perform their function. And again, it will help CD8 cell in killing as well. So it's, if you see, mainly in all these parts, here is interferon gamma, here is interferon gamma, here is interferon gamma. That's the key. So TH1 is differentiated because of IL-12, but the orders by CD4 are given through interferon gamma. So far, so good, no? You look all exhausted. If there is any confusion here, you can ask. Yes, please.
CD8 can't work without CD4?
It can, but so if you remember, CD8 cell have got CD8 receptor which can directly recognize MHC class 1, but that will be after activation. That's number one. Number two, as I said, also, this CD4 is like a pushing, it's like a supervisor who can push everyone. So he will push CD8 as well, that kill more. Because the feedback system. So if there is lot of virus, that feedback mainly comes to the CD4, and then CD4 tells that, okay, start pushing, kill more. But if not enough viruses, he will not push. So CD4 is actually at the center of everything. It will tell B cell, it will tell macrophages, it will tell CD8 cell to perform functions efficiently. So the general rule is, when TH1, it is called as cell-mediated immunity. Now, one clinical example I'll give you here, delayed type hypersensitivity. Have you heard of this before? So I think by the end of this week we have got TB, TBL. Yeah, so that is through, there is delayed type hypersensitivity. Why? Have you studied tuberculosis so far?
No.
No. Okay, so Mycobacterium tuberculosis is an intracellular pathogen. Okay, we have talked a lot about intracellular pathogen. Which cells will be activated against it? CD8. And I told you, one example I gave you of virus, that he's very tricky. He sometimes switches off MHC class 1. Mycobacterium tuberculosis is also very tricky. He sits inside the macrophage and does not allow the phagosome to join the lysosome. So when it does not join, it will not release the enzymes, and the killing mechanism will not take place. So it kind of prevents the phagosome lysosome joining and just stay inside the cell for months, and as long as it can stay there, it hides there. That's why lots and the T cell thinks that why this mycobacterium is not finished from the body. They keep on producing interferon gamma and keeps bringing more macrophages, more T cells, and more and more recruitment. Come, we have to kill it. And there mycobacterium is sitting inside the macrophage just trying to avoid its death. And as a result, what happened? Lots of cells are there now. This is called as granuloma. We'll discuss more in the TBL, but just for now, what you need to remember is that mycobacterium is a tricky organism. It stays in the macrophages, prevent its phagosome lysosome joining, and as a result, T cell has to bring in more and more cells, and as a result, a big giant infiltration of cell is produced that is called as granuloma. And this you can test in the patients. There is a test which is called as tuberculin test. You can, they have small pricks, you put it on the skin and then encircle it, and you read it after 72 hours because there is a delayed response. It takes time to react, and then there is induration, red coloring of the skin. So this is based on Th1 mechanism. So when the body has helper T cell, they produce interferon gamma, they call in all these cells, immune cells, they will result in this tuberculin reaction. Number 2 is activation and function of macrophages. I've already talked many things about it, that CD4 will help in the activation of macrophage through interferon gamma. And if you look, what is the main difference between the macrophage, the first macrophage and the second macrophage? What are the differences that you can see? So there are two, size. What else? Lot of receptors. What else? Do you see any words written inside the cell? ROI, reactive oxygen intermediate. And what is else? Nitric oxide. So these are the differences. Normally, when macrophage is not activated, it is not large in size, it will not produce reactive oxygen intermediate, it will not produce nitric oxide, and the receptors on its surface, main receptors and co-stimulatory. So all these things, now you can see here, this is the explanation. Increased co-stimulatory receptors is done by interferon gamma. Number 2, secretion of cytokines. So this macrophage will start producing TNF, IL-1 and IL-12. It will increase expression of MHC receptor and more killing ability with the help of ROI and NO. Now we'll quickly go to Th2. Which is the cytokine for Th2? IL-4, very good. And this IL-4, so remember, Th1 is cell-mediated immunity. Th2 is humoral immunity, antibody-mediated, okay? And against two main things, allergy and parasites. So when you think of parasites, you should know eosinophils. Now in this figure, you will see that IL-4 has resulted in, number 1, class switching. It was producing IgG, it will now start producing IgE as well. And IgE is the main antibody in allergy. So anyone who is allergic to peanuts, to milk, to different kind of dust particles, to cat hair or to dander, they will have increased IgE in their body. So if you will measure their immunoglobulin level, IgE level will be very high. Number 2, if somebody has got parasitic infection, again their immunoglobulin E as well as eosinophils, they will be high. So here you can see that the general rule is, if Th2, you should think of humoral immunity. Two situations clinical that you should remember: allergy involve IgE and Th2, and asthma. Asthma is also, there are different types of asthma. So the allergic asthma, I'll give you a lecture on that as well. So we'll discuss allergy in hypersensitivity, and on asthma there will be, in the next module lecture. So just you remember, Th2 is linked with allergy and asthma and parasitic infection.
What does it suppress?
Okay, normally whenever there is a Th1, I think there are a couple of slides about that, but I'll explain it to you now, that in the body you have Th1 subset, you have Th2, you have Th17. If I take out blood from any one of us, we will have all these cells. Any one group will be in domination. So one group will be strong, others will be little less. Now, if Th2 is working, then Th functional part, which is CD4 and macrophages, they will not function very well, because at one time when IL-4 comes, it suppresses IL-12. When IL-12 is working, it suppresses IL-4, so vice versa. So one group works, the other calm down. When the other works, IL-4 drops down. So among Th1, Th2, Th17, one is going to function, the other ones are going to calm down. Otherwise, they will interfere with each other functions. Okay, so one work at a time. Now in Th17, it's mainly IL-17 is the cytokine for this, and this is mainly involved at the mucosal surfaces. Two clinical conditions that I will discuss now is rheumatoid arthritis. Primarily, it's an autoimmune diseases of the small joints of the hands. It's symmetrical, both hands or both sides of the body are involved, and then it also involves lungs, kidneys and skin, but mainly disease of the joints, and it is because of the domination of Th17 cells. Another disease is inflammatory bowel disease, IBD, which has got two variants, Crohn's disease and ulcerative colitis, again an autoimmune disease of the GI tract. So in this condition also, Th17 is found to be involved, and we'll discuss both these later. So there will be lectures on these. Now finally, we have found out that Th1 works against cell-mediated immunity like tuberculosis. Th2 works in, against parasites and allergy or asthma. Th17 works in autoimmune conditions like rheumatoid arthritis and IBD. But regulatory T cells, they are there in our body. They are produced by the thymus, and they stay in the body to keep a check and balance. If these immune responses keep happening, they will destroy all the tissues. So once the elimination of bacteria or a parasite has occurred, these regulatory cells have to inhibit them, stop them. That's why their name is T regulatory cells. What is special about them? They have got CD4, but with that another extra receptor, CD25. So they are CD4 positive, CD25 positive, and their main job is to inhibit unnecessary immune reaction, or when the job is done, to stop the immune response. Now this is the picture. It's in a healthy immune system, you will have Th1, which is blue, you will have T17, which is purple, you will have Th2, which is pink, and then you will have T regulatory cell, which is green, and they will all need to be in balance. Okay, but if this balance is lost, what happens? Now there are different conditions. If first one, where the pink cells are more, Th2 is dominating, while the two Tregs are less, and there is no Th1, this is the example of allergy. Okay, here it is written asymptomatic atopy. Atopy is allergy. Then if you see furthermore, Th2 and Th1 are there are no regulatory cells, then they start to have allergic disease. It's all about the balance between these cells. If Th2 is more, the patients are going to have allergy. If the Th1 are more, the patients are going to have autoimmune diseases. And the last one you can see again, if Th1, Th2, Tregs, they all are together in a balanced number, then the patient is healthy. And here again, you know, the point that I already talked about, that Th1 produce interferon gamma, Th2 produce IL-4, and IL-4 is going to inhibit the function of macrophages. So at one time only one will work. And you can see here in the second figure that IL-4 will inhibit Th1, interferon gamma will inhibit Th2. So they have inhibitory effect on each other. And this again, this figure is same. We will see one by one that if Th1 is active now, its interferon gamma is going to inhibit both Th17 and Th2. If Th17 is active, it's going to inhibit Th1, and finally Th2 is active, it will inhibit Th1. So one stay active, the other one stay calm. So any questions? Anything that was not clear? Yes, please.
B cell work in intracellular, extracellular.
B cell mainly works in extracellular pathogens. That's, you know, their main function is to work against extracellular pathogens.
What is the third mechanism of CD8?
What is the worry?
The third mechanism of the CD8 to kill the
Death receptors. So the fast ligand and the fast receptor, whenever they join, they will again also trigger the apoptosis. Yeah.
What is IL mean?
Interleukin. You remember I told you that the cytokines, they help in communication of the immune cells with normal cells, but you see how exciting is immunology. So he's asking what is interleukin. Inter is between, leukin is white blood cells, those cytokines that helps the white blood cells communicate. One example is interleukin 4, interleukin 7, interleukin 5, you know, among what we have seen today. Interleukin 17. So why they have these numbers? So there are hundreds of them, you know, so far discovered. As they discover, they just give them a number, and then later they associate their functions. It can do this. This is the function. This is the function. You didn't get it. Okay. So, you know, in the previous slide we talked about how I will want you to remember this whole concept. First thing is that CD4 cells can polarize. They can become Th1, they can become Th2, they can become Th17. Now this, as you grow older, somebody has infection with one type of bacteria, another type of bacteria, somebody has infection with a parasite. So as you grow older, your Th1, Th2, Th17 memory cells will be different. We all have different exposure of bacteria and infections. So our T cells exposure and their presence in the body is different. So one thing is that which cell is during an immune response, whether you will have Th1, Th2 or Th17, that is one thing. Another thing is that right now we all are healthy, alhamdulillah. If I just check any one of us immune cells, Th cells, whether we have Th17 more, Th2 more or Th1 more, we will all have different ratios. And that is because of what exposure you had in your previous life. Okay, and which kind of environment you are living with. Now I'll give you one example. It will make you, understand this, that the West has got maximum incidence of allergic diseases. Th2. Why? Because of the hygiene hypothesis, which means that the more you are very, very clean, one is normal, observing good hygiene, washing your hand before eating or this. One is extraordinary hygiene, those anti-disinfectant creams and lotions all the time, and that will result in, when you are in too much clean environment, that will shift the Th1 or Th2 balance. And they found that the more you are in very extraordinary clean environment, you are more prone to develop allergies because of the imbalance. This is called as hygiene hypothesis. So it is actually sometimes better to be exposed to these different bacteria, parasites. I'm not saying to have infection all the time, of course, just a normal environment. Don't try to be any extreme, extraordinary clean, extraordinary dirty. There is a balance, always the medium is the best. So with this, there will be a balance between the Th1, Th2, Th17 subset. Now coming to the immune response. Whenever there is an immune response to any of these, to extracellular bacteria, it means it is Th2. To intracellular bacteria, it means Th1. To mucosal infection, it means Th17. Now, whenever there is infection, there will be polarization of one type, whether Th1 or Th2 and Th17, and it will not allow the others to work. So example, extracellular bacteria come, Th2 is activated, it will not allow Th1 or Th17 to work. It will work itself, finish the job, and then calm down. So that during an immune response, only one work. Normally, we have a subset of Th1, Th2, Th17, and their balance is actually healthy. Is this okay?